So first off, next week on Monday, we're going to meet in person and have a lab day. And then on Wednesday, we might do a little more lab stuff. And then we will also do our test on chapter 7 and 8 on Wednesday. So in-person days next week, I believe the 17th and the 19th, Monday and Wednesday next week. So I think probably today we'll finish up cellular respiration. And I just wanted to, review a little bit of where we have been, because I know this is one of the more difficult units that we'd go through. Let me find my slide that I had here. Okay. So cellular respiration is basically the metabolism of glucose, catabolism, more specifically, the breakdown of glucose. And we break that glucose down to get ATP energy for our cells. We need a constant supply of that ATP energy, otherwise the cell dies. And that's true from all the way down to prokaryotic unicellular bacteria, all the way up to like elephants and plants and fungus and humans and all of that kind of stuff. All cells need ATP to survive. Okay, so the main purpose of cellular respiration is to produce that ATP. Now, within cellular respiration, or this catabolism of glucose, we have several different stages, and each stage is a different metabolic pathway. Okay, so each stage is a series of chemical reactions. And so we start out cellular respiration, and this should be reviewed from last time, with one glucose, which has 6 carbons. Okay, through step 1, or I guess you could say stage 1, or pathway 1, you have glycolysis. And we said that lysis means to split. Glycolysis means splitting of glucose. So we learned last time that at the end of glycolysis, we split 6 carbon glucose into 2 3-carbon pyruvates. Okay, so we started out with 1, 2, 3, 4, 5, 6. We split that in half, and now we have 3 carbon pyruvates, 2 molecules of 3-carbon pyruvates. Also, throughout that process, we saw that we get a net of 2 ATP. And if you remember back, glycolysis has that investment stage where we have to invest a little money or invest a little ATP to get the process started. So we invest, or we put in 2 ATP. We get 4 total produced, but since we already used up 2, we say that we have a net 2 gain of ATP. We also, and this diagram isn't showing it, the other thing that we get from glycolysis is NADH. And remember, NADH is those little Ubers or electron carriers, and it carries high energy electrons. And all of the NADH go to step 4, the electron transport chain. Okay, now after glycolysis, if there's oxygen present, like in our cells, okay, pyruvate is going to go to step 2, which is acetyl-CoA formation, or sometimes called this acetyl-CoA synthesis, or the prep step. And then step 3 is the Krebs cycle. And step 4 would be the electron transport chain. If there's no oxygen present, and we'll talk about this at the end of the chapter, Pyruvate can then enter what's called fermentation. And it just kind of breaks down the pyruvate a little bit further in a different way. So last time we talked about steps 1, 2, and 3. Acetyl-CoA formation pulls off a carbon, and you lose some of those carbons as CO2, and you also get some NADH. And then the final product of that is acetyl-CoA, which goes into the Krebs cycle. And by the end of the Krebs cycle, remember we had 6 carbons in glucose. All 6 carbons that were in glucose originally are going to be lost as carbon dioxide. And through the Krebs cycle, also you get NADH, electron carriers, you get FADH2, you get some ATP out of the Krebs cycle, but these electron carriers are going to go to the electron transport chain. Okay, so that's kind of where we left off, is at the end of the Krebs cycle. And so we'll jump ahead and kind of pick up where we left off. Okay, and this slide, I believe, is the last one that we filled in, and it's basically saying, you know, at the end of the Krebs cycle, you know, we've made only a few ATP, but we have all of these high energy electron carriers, okay, or electrons in the carriers. And so the electron transport chain is going to be really where you get most of that ATP produced. Okay, we get a little along the way, but it's not until basically step 4 that you get most of the electrons. Okay, so it says in the second, which I'm not using their steps, in the last step of this process, high energy electron carriers, which are going to be your NADH and FADH2, release 2 electrons into the electron transport chain proteins. Basically, and we've kind of used this analogy before, NADH is the Uber that picks up the passenger at the airport, and now they're going to drop off the electron passengers at the hotel, for example. Okay, at their final destination. And so the electron transport chain is really just a series of really proteins that are in the mitochondrial membrane. And then before I bring up the image, I guess, the depleted carrier, so the empty Ubers, so this would be NAD plus and FAD plus. The empty electron carriers, again, are now available for recharging by glycolysis and the Krebs cycle. So they can be recycled. So they're basically, you know, like those little shuttle buses. They pick up people at the airport parking lot. They drop them off at the terminal. They go back to the parking lot. They pick them up, drop them off at the terminal. And it's the exact same thing as like a shuttle bus driver. All they do their whole entire life is pick up electrons, drop it off at the electron transport chain, and then go back in those earlier steps. Okay, so I'm going to pull up, you guys have the little worksheet that looks like this. I'm going to pull that up. I really like these. I like how they're set up, and you guys can write on them. So go ahead and pull that out. I'll give you a second to grab it. On the top, it says oxidative phosphorylation. Okay. So to kind of orient a little bit here, I'm not sure why we have this here. Move out of the way. Okay, I don't know. Anyway, sorry. Okay. So to kind of orient, remember in eukaryotic cells, we're going to be in the mitochondria. In prokaryotic cells like bacteria, we are going to be in just the cell membrane. But you can see here, all over around here is going to be the phospholipid bilayer. Here's another phospholipid bilayer. And then all these little structures here in the center are going to be different proteins of the electron transport chain. So there's 1, 2, 3, 4 large proteins here that come together to make letter R, which is the electron transport chain. Okay, so that's what we're looking at there, is the electron transport chain. And then we just said in the previous PowerPoint slide that that's going to be embedded in the inner mitochondrial membrane. So this here, letter K, is our inner mitochondrial membrane. Okay, and so remembering mitochondrial structure right now is kind of important. Okay, so letter R is the electron transport chain. All of these structures, these proteins here embedded in that inner membrane is the electron transport chain. And then the phospholipid bilayer that it's a part of here is the inner mitochondrial membrane. So then, of course, I bet you guys can figure out what J is. If that's the inner membrane, probably can figure out what J is. And then this space here between the membranes, okay, that is letter P, and that's going to be the intermembrane space. And they don't have it, I'm not seeing it. Oh yeah, they do. Never mind. It is listed. Letter L over here. Okay, letter L refers to just this whole space out in here. And that's the mitochondrial matrix. And it's very, basically like the cytoplasm or the cytosol that fills the space inside the cell. This matrix fills the space inside of the mitochondria. Okay, so I'm going to clear all my little squigglies there. And I was kind of geeking out here last night, actually, as I was reviewing for your lecture. And I was reviewing some of my biochemistry. And each one of these, one of these proteins that we're going to talk about and actually do a case study in reference to is this guy here. And this protein, I'm not going to make you remember this. I'm just geeking out with you right here. This is called cytochrome C oxidase. And remember, ASC at the end of the word means an enzyme. But each of these proteins here in the chain, they actually have different names and all of that. But later on in some of our work that we're going to do, we're going to talk about cytochrome C oxidase. And its job is to basically take up the used electron and pass it on to oxygen. Okay, but we'll talk about that a little bit more later. Okay, so if we look very closely in here, letter T is our NADH electron carrier. So remember, electron carriers, and I'm beating this dead horse because I want you to for sure remember it. These all come from the other steps, right? Glycolysis, Krebs cycle, whatever. All of those electron carriers that are produced there come here. And so T is NADH, nicotinamide adenine dinucleotide. And you can see over here, it's very tiny, but you can see that it's dropping off its 2 high energy electrons that it had plucked out of glucose or its little intermediates as we've gone through the previous steps. And remember, electrons cannot just be released out into anywhere, right? These electron transporters have to carry them from place to place because they can't just be released. Okay, it's like they have to be passed along, otherwise they cause a lot of damage to the cell. Okay, and so as these high energy electrons are passed through this protein, some of the energy from the electron is used to pump hydrogen ions into the intermembrane space. Okay, so we've taken high energy electrons from glucose pieces, and we've taken that energy to pump hydrogen ions where you have a high, sorry, my pen isn't really working here. Electron constant, oops, sorry, not electron, high hydrogen ion concentration out here and a lower concentration out here. So what you're doing is it's basically active transport. You're pumping a bunch of hydrogen ions from a place where there's a lot of them into a place where there's not as many. And what you're trying to do is you try to build the hydrogen ion concentration in here. Okay, so as the well, and I guess if we want to look down at the bottom, letter V is the hydrogen ions. Okay. Then this electron from here is then shuttled to another protein. And then also over here, FADH2, which is letter U, flavin adenine dinucleotide. FADH2 drops its electrons off over here at the second protein. Okay, it needs a special drop-off place. So for example, NADH is going to the United terminal, FADH2 has to drop off at the Southwest terminal, okay? But then they both get transferred to this third protein where again, both of those all of those electrons are pumping more hydrogen ions in. They get passed to cytochrome C oxidase, where we pump in a bunch of hydrogens here. And from here, the electrons are passed to oxygen. Okay, so X is oxygen. W, of course, is water. And then what happens is you get this huge buildup now of hydrogen ions in the inner mitochondrial membrane, or I'm sorry, in the inner space. And just like we saw with the Calvin cycle, ATP synthase, letter Q, which is this protein over here, Q then allows the hydrogen ions to go from a high concentration here to a lower concentration out here. Just like water flowing through a turbine with hydroelectric power, that those hydrogen ions spin, that flow spins the enzyme, and it allows the enzyme to have the energy to add a phosphate onto a DP and create ATP. So S, of course, is ATP. Okay, so in a nutshell, that is basically what happens with the electron transport chain. Same exact type of process that we saw as part of photosynthesis in the light reactions. And we're also using, remember we had that term chemiosmosis. Okay, we'll get back to that when we kind of get into the notes and stuff. But this is essentially what's happening here. Okay, and so the technical term, we call it the electron transport chain, but in higher levels of biology, biochemistry, they call it oxidative phosphorylation. And phosphorylation, just to geek out on you, is basically like when you phosphorylate something, you add a phosphorus to it. And so basically what you're doing, it's like how ATP is made. Phosphorylation is how this ATP is made. And so that's just the technical term of what's happening in this process. Okay. Hang on to these. We're going to switch back to the notes here for just a little bit, and I'm going to have you work on some more of this worksheet. Close that out. Okay. So here's just another look at the same exact process, just in a different format. I do like that they have the mitochondrion up here, and you can see where that's happening. So they're just looking at right here is just the inner membrane, whereas before we had the outer membrane that we could see out up in here. And then on the inside, we have again the matrix, and we have all of those proteins that are involved in the transport chain. Here's ATP synthase, where the ATP is made. This is cytochrome C oxidase. You can see where NADH is dropping off its electrons and pumping hydrogens. And I just noticed they have the arrows going different ways. That's interesting. Anyway, so then we pass the electrons around, and hydrogen ions are pumped across the membrane. You build up a concentration. Actually, they don't have it backwards. Never mind. And then you get the ATP produced as those hydrogen ions flow through here. It gives the energy through chemiosmosis to add or phosphorylate that ATP. And then what happens here is that oxygen accepts the electron, and it's combined with some of those hydrogen ions to create water. Okay, and one thing that I really want to point out here is that this is kind of like a bucket brigade. And if there's no oxygen present to pick up these electrons, what happens is this chain becomes saturated with electrons, and then the NADH becomes saturated with the electrons. So think of it as like they closed the doors on the terminal of the airport. Okay, so none of the passengers could get out of the shuttle bus. So eventually all the shuttle buses would be filled with passengers and they would just be sitting there. Okay, and in this situation, like, the passengers can't get out of the shuttle bus, they are trapped in there. Okay, and so you still have people coming into the parking lot and people trying to get into the shuttle buses, but eventually all the NADH shuttle buses and all the FA22 shuttle buses become saturated and filled, and basically the whole chain backs up and ATP production stops. And if that happens significantly, for a significant period of time, that cell is going to die. Okay, and this is why if we cannot get oxygen, if we cannot breathe, our cells start to die, because we have all of these electrons stuck throughout the chain, and ATP can't be produced because these hydrogen ions are no longer pumped across the membrane. Okay, so that's why when a person suffocates, they die, because they cannot continue cellular respiration. That shuts down that whole metabolic pathway. Okay, so let's go on in our notes here. So the electron transport chain in the mitochondria is basically the same function that we saw in the thylakoid of the chloroplasts. Job is to make ATP. And we saw high energy electrons jump from protein to protein, from molecule to molecule, releasing their energy along the way. And if we go to the next slide, that energy is used to pump hydrogen ions from the matrix to the intermembrane space, producing a concentration gradient of hydrogen ions. Okay, so producing a high concentration of hydrogen ions. And then this hydrogen ion gradient is used to make ATP through that process we talked about, chemiosmosis. So, as we saw a little bit ago, oxygen is the final electron acceptor in aerobic cellular respiration. Okay, it is essential. Without it, there is no ATP production produced by the electron transport chain. At the end of the electron transport chain, all those depleted electrons, they were high energy, now they're pretty low energy. They're going to be all transferred by cytochrome C oxidase to oxygen. And this is what we call the final electron acceptor. These energy depleted or low energy electrons, the oxygen and hydrogen ions that are already in the mitochondria, combine to form water. And one water molecule is made for every 2 electrons that pass through the chain. So if no oxygen is present to accept the electrons, the chain becomes saturated with electrons, and cannot get more electrons from NADH or FADH2. Okay, the hydrogen ion gradient would dissipate or basically disappear, and ATP synthesis by chemiosmosis would stop. ATP production continues only when there's a steady supply of oxygen. Now, at the beginning of this chapter, we talked about electron transport chain producing around 32 ATP. You only get 4 ATP from the rest of the 3 steps. And so if you eliminate this major source of ATP by eliminating oxygen presence or final electron acceptor presence, that is very, very harmful to the cell. Okay, so without oxygen, or you could also say the final electron acceptor, ATP production drops and the cell can die if that's a prolonged period of time. So basically a human can only live a few minutes without oxygen, because within that few minutes cells start to die. And when a significant number of cells in an organism die, then that organism also dies. Okay, and we're gonna look at that a little bit as we go through our case study that we'll do a little bit later. Okay, so chemiosmosis, we've already talked about this. Basically, just to define it again, is the process by which some of the energy from the concentration gradient is captured in ATP. So basically, it's the production of ATP using flow of hydrogen ions from a high concentration to a low concentration through ATP synthase. And remember, ATP synthase is an enzyme that is capable of adding or phosphorylating, adding that phosphorus to ADP to make ATP, adenosine diphosphate. So again, that's just the basically what's happening with chemiosmosis, and it's just a term to describe that part of the electron transport chain step. Okay, so hydrogen ions flow across the membrane through, like we've said, ATP synthase. And that basically makes the equation that we just had on the last slide. So glycolysis and cellular respiration, which they're calling steps 2 through 4, acetyl-CoA synthesis, Krebs cycle, and electron transport chain. All of that produces obviously more ATP than glycolysis alone. The whole process, steps 1 through 4, make 36 ATP per glucose. Okay, Star that, you must know that. Okay, so for every molecule of glucose that comes into this catabolic process, 36 ATP are produced. Okay, and so like I said a little bit ago, you're only getting 4. Here's the electron transport chain down here. You get 2 net here, 2 net here. You get 4, and then you get 32 from the electron transport chain. So if down here, oxygen is necessary, glycolysis can happen with or without oxygen. But if we do not have oxygen, okay, basically this stops, the electron transport chain stops, and you start to get things backing up here as well, and it eventually will back up and stop those processes as well. And so kind of depending on the book, some books say that, you know, oxygen is necessary for all of those, because if you don't have it at the end, everything backs up and all of this stuff stops. But glycolysis does not need oxygen because, like we said at the beginning of today, these pyruvate can go over to another process called fermentation. So glycolysis can continue even if there's no oxygen present. Okay. This is all really squiggly. Let me erase all of this for right now. Another thing that I wanted to point out, and let me make sure I'm not getting ahead of myself. Okay, so we were talking about aerobic cellular respiration, where oxygen is the final electron acceptor. Okay, so as those electrons are passed through, oxygen's our final electron acceptor. Okay, and this is true for all plants, animals, and fungi. I'm not 100% sure about protists, but there are many prokaryotes, bacteria and archaea, that are anaerobic. There are some microbes like Clostridium tetani that causes tetanus, or Clostridium botulinum that causes botulism, that are actually unable to survive in oxygenated environments. Okay, so they undergo what's called anaerobic cellular respiration, and the process is very similar. They have some different proteins in their electron transport chain, but their final electron acceptors are things like nitrate, sulfate, oh shoot. I want to say carbon dioxide, but I'm not sure about that one. But they have other things that they use as final electron acceptors. Okay, so when we talk about aerobic cellular respiration, we have to have oxygen, but in these anaerobic microbes like botulism and tetanus, they have to have one of these as well. They would die without their final electron acceptor. Okay, so just because it's not oxygen, they still have to have one of these to pick up the electrons to continue along the chain. Okay, so remember, as we're approaching the test next week, some things to keep in mind for, well, really cellular respiration and photosynthesis, but excuse me, what is the reactant? Okay, So we talked about the equation for cellular respiration. Okay, and we have some reactants and products there, but what it more specifically, so you'll need to know that, but more specifically, what are the reactants and the products for each of the 4 steps? Okay, so for glycolysis, the reactant is going to be glucose, and your products are 2 3-carbon pyruvates, 2 NADH, which notice the arrow goes all the way down to the electron transport chain, and 2 ATP. Okay, and so anytime we see ATP produced, just remember that it can be used immediately by the cell. That ATP there could be used immediately to start glycolysis again, and another glucose. Okay, so assuming oxygen is present, those pyruvate will go into step number 2, which is acetyl-CoA formation. And during acetyl-CoA formation, that pyruvate is going to lose a carbon and produce NADH. Now remember, we have 2 of these pyruvate. So this one's going to go into acetyl-CoA synthesis and then into the Krebs cycle. Then this one is going to go into acetyl-CoA synthesis and then into the Krebs cycle. So acetyl-CoA synthesis and the Krebs cycle happen twice for every glucose molecule that enters the process, enters the metabolism or the pathway. Okay, so for 1 pyruvate, you get 1 NADH and 1 carbon dioxide. And then for the second, you get the second NADH and the second carbon dioxide. Okay, so from here, we lose one of these carbons. And now acetyl-CoA is basically 2 carbons and then the CoA molecule added to it. This acetyl-CoA goes to the Krebs cycle. This acetyl-CoA goes to the Krebs cycle, and by the time both of them have gone through, you get 2 ATP. The remaining carbon dioxide or remaining carbons are lost as carbon dioxide, and then you get these high energy electron carriers that go to the transport chain. And we just talked about that, and they get 32 ATP produced from there. Okay, now, again, this is kind of geeking out a little bit, but we talk about what's called theoretical ATP production. And so technically, they would say a eukaryotic cell has a theoretical ATP production of 32 per glucose. Okay. I'm sorry, 36. In actuality, this is around probably 30 to 32. Okay, so the 36 is like a book number. Okay, so like if everything happened perfectly, the most you could get with a eukaryotic cell is 36. In actuality, there's probably only 30 to 32, and let me show you why. Some of these hydrogen ions here can sneak back across if you have problems with the membrane. And some of these hydrogen ions, like if the pH of the cell is a little bit off, some of those get taken up into buffers, like bicarbonate. And so not all of those hydrogen ions are actually used to run ATP synthase. So there's a little bit of inefficiency there. And so in actuality, the cell probably is only going to get to make 30 to 32 in a real life situation. But for textbook purposes, you know, if I ask you, what is the ATP synthesis of or production, what's the total number of ATP per glucose throughout cellular respiration, and you tell me 36, that's the number that I'm looking for. But I just wanted you to know that in actuality, it's a little bit lower. The other number that I wanted to point out here is that this is for eukaryotes, like us, plants, whatever. Prokaryotes, the theoretical ATP production is 38. And you're like, well, what in the world? You just told me like this whole process is exactly the same when you use aerobic cellular respiration between bacteria, humans, whatever, when it's aerobic, when it's in the presence of oxygen. Yes. But you lose some ATP there, about 2 or so, when you have to take the pyruvate and get it into the mitochondria. That has to be pumped in, and eukaryotes don't have to do that. I'm sorry, prokaryotes don't have to do that because they don't have a mitochondria to pump it in. And so prokaryotes get a little bit more ATP from this process than eukaryotes, just because they don't have a mitochondria to get that pyruvate into. And then anaerobic cellular respiration, their theoretical, it ranges between around 2 and 38. And it just really depends on the final electron acceptor, the proteins they have. It just really is variable for anaerobic cellular respiration. Okay, so anyway, that's my little geek out moment there for you guys. Okay, so we've seen this chart before. You guys have filled it in at the beginning of this chapter there. So glycolysis, I don't know why this did that. That's kind of weird. I think it did it in the other chart too. But glycolysis gets you 2 NADH, 2 net ATP. Acetyl-CoA formation in the Krebs cycle gets you 8 NADH and 2 ATP. Happens in the matrix. That's the other thing you need to remember, is where these reactions are occurring. Then step 4, electron transport chain. These are released. Okay, these are not made here. These travel to the electron transport chain and help make those 32 ATP. And then we'll talk about fermentation here in just in a little bit. And we get our total of our theoretical ATP. All right. Show this little video clip.
Electron carriers such as NADH deliver their electrons to an electron transport chain embedded in the inner membrane of the mitochondrion. The chain consists of a series of electron carriers, most of which are proteins that exist in large complexes. Electrons are transferred from one electron carrier to the next in the electron transport chain. Let's take a closer look at the path electrons take through the chain. As electrons move along each step of the chain, they give up a bit of energy. The oxygen you breathe pulls electrons from the transport chain, and water is formed as a byproduct. The energy released by...
Oh, sorry. Got too excited. Shoot.
Hydrogen ions, the blue balls, energy...
Okay, pause. See how those electrons are just waiting there? Okay, they can't leave until they're picked up. Okay, and that just kind of further shows you how if there's no oxygen to pick these up, everything backs up further up the chain.
The energy released by electrons is used to pump hydrogen ions, the blue balls, across the inner membrane of the mitochondrion, creating an area of high hydrogen ion concentration. Hydrogen ions flow back across the membrane through a turbine. Much like water through a dam, the flow of hydrogen ions spins the turbine, which activates the production of ATP. These spinning turbines in your cells produce most of the ATP that is generated from the food you eat. The process you've just observed, cellular respiration, generates 10 million ATPs per second in just one cell. That ATP can power a biker up the trail, or it can power your brain cells as you learn challenging biology topics.
All right. Okay, so let's give your brain a break. I've got 8:53. Let's come back a little before 9. And we'll kind of wrap up and look at fermentation, kind of wrap up chapter 8. Okay, so let's take maybe about 5 minutes, and I'm gonna have you guys answer on your worksheet questions 13 through 16. And then on number 17, it says many of the poisons kill living things by inhibiting or stopping or slowing down essential components of oxidative phosphorylation, or basically the electron transport chain. It says use the internet to look up uncoupler or uncoupling agent, and you'll find some poisons. Pick one and kind of tell me a little bit about how it works as far as affecting the electron transport chain and ATP production. So I'm not looking at like a 100-page essay or anything like that, but just in a couple of sentences, tell me how it works, and go ahead and answer those questions. I'll give you guys a few minutes for that. If it takes longer, you can always upload it later.
I just looked at, I don't jump in online for one of them. That has the pharmacology, withdrawal, like how. I'm doing like. That's just which, as long as it has all the information you need, like when you scroll down. You can see that. Session. That's awesome. Okay, this is what environment carries when it comes to toxicity.
Whenever it says, where can it be found? Mine is just manufactured, so
That's fine.
like mine says it can be found in sweet clover, that's fine.
Yeah, yep. Okay, so let's take a minute or so, kind of finish up the thought that you're on, and then while we're finishing up that, we're gonna start a Kahoot and kind of quiz you guys a little bit about cellular respiration. And so I'm gonna pull that up if you want to finish up your thought on the worksheet and then just join us. Go to kahoot.it. And then we'll have our pin here in a second.
I had to agree with my because I was copying the famous state, why is this true? Mine is for like one plus though. So that's why mine is dangerous. Is it for you? That's what you have to ignore. I'm busy. I was taking a minute. Bro, fine, it's fine. We're gonna get over what we just went through. So just try to get this.
Is there a Dropbox for that worksheet?
Yes.
Okay. Yeah, because it's gotta be really wrong.
All right. Ready? Here we go. Yep, the reactant, the molecule that goes into the step of glycolysis is glucose. Okay, I think we're stuck. Shoot. Hello. Uh-oh, did I kick you out? Oh,
Yes.
dang it. I'm sorry. My computer froze. Okay, we'll try again. Okay, you should all get this one right. Hooray. Are we stalling out again?
Next button on the top right.
Oh, it's hiding. I'm sorry. Thank you. My first day. Good. ATP invested during glycolysis is 2. We have to put in 2 to get the process going. Good job. Oh, tricky one. Glycolysis is in the cytoplasm. After that, we move to the mitochondria. Good. 3 carbons in pyruvate.
That's my question.
Good. Good job. Goes to acetyl-CoA formation.
Good job.
If there is oxygen. If there's not, it will go to fermentation. True or false? Oh, true. Yes. Carbon dioxide is produced because we pluck out one of those carbons from pyruvate, and then you also get NADH. Yay. True. All of the carbons that come in on glucose are eventually lost as carbon dioxide. Woohoo! Good job. Yep, acetyl-CoA enters the Krebs cycle. Brianna's on fire.
Check. Oh, heck no.
Sorry, I forgot to take off the check all that apply because you have to like pay money for that one. So I took it off and just put in all of the above, and I forgot to change the question. But yes, all of these are products of the Krebs cycle: carbon dioxide, ATP, and FADH2. Yes, I've been beating that dead horse for 2 class periods, and you got it. Yay, good job.
I got about a lose. It's last question.
Okay, this was more of a, I wonder which one they feel like is the most confusing. So good. I didn't include the electron transport chain. We'll include that in another Kahoot on Monday, but good to know.
I don't know how.
Yay, that was pretty close. Good job, guys. All right. Okay. Okay, so let's switch back. And so let's finish up chapter 8 and talk a little bit about some fermentation and things like that. You know, let me skip to this slide here really quick first, just to give you kind of a visual of what that other slide with all the words is talking about. So when we talk about cellular respiration, we use glucose as an example when we teach this, because as you can see here, glucose goes through all of the steps, okay? So glucose gives us an example of the whole entire process. But we know that we don't just eat glucose, right? This taco here, it's getting kind of lunchtime, probably, well, no, actually it's only 9:30, but maybe you're hungry for tacos. And, you know, tacos have got the carbs in it from the tortilla or the shell, whatever. It's got some protein with the meat and the cheese. It's got some carbs as far as, you know, the vegetables and things on there. It's got some fats with the meats and cheeses and probably the tortilla as well. And so it's important to know that, you know, just because we talk about glucose being used to make ATP, we also know that there's calories in proteins and fats too. And so the reason that we don't spend so much time talking about proteins and fats when we break them down is because they enter at various points. So let me back up here just a little bit and talk about the digestive system for a second. So when you eat food, okay, you do what's called mechanical digestion, which is an example of that would be chewing, and you also do chemical digestion, okay? So remember, glucose is a carbohydrate. And a lot of where we get glucose from in the diet would be other carbohydrates called polysaccharides. Remember that term? We also have disaccharides like table sugar, which is glucose plus a fructose. A polysaccharide would, and in this example, would be starch. Okay, well, I was gonna write starch there. Well, maybe I'm not. Let me try this other pen. I was gonna write starch there. We'll try this here. Okay. Well, let's try to do this then. Good grief. Okay, so starch, remember, is a big long chain. It's a polysaccharide with a whole bunch of glucose molecules, like hundreds or thousands. Okay. And this takes us back to our biomolecules. Proteins, remember, those are chains of amino acids. Okay, amino acids are the monomers, the protein would be the polymer. Fats are, remember, a triglyceride where we have the glycerol. And we have the 3 fatty acid chains that attach to it. Hopefully this brings back memories from like chapter 3. Okay. In the digestive system, in order to get the food or the nutrients out of the digestive system and into your bloodstream, which carries the nutrients around the body and delivers them to the cell, in order for that to happen, that process is called absorption. In order for that to happen, your digestive system has to break apart these individual monomers. Okay, we have to do hydrolysis reactions to break down starches into individual glucoses, break down proteins of cheese into individual amino acids and to individual fatty acids when we're talking about the lipids. Okay, so when we do that, glucose can go all the way through this process. Okay, I'm going to clear this really quick. But also when we have amino acids from the cheese or the hamburger or whatever, depending on the type of the amino acid, some of them can be converted into pyruvate and then enter and go through the rest of the cycle to make a few ATP. If they entered here, they would end up making 34 ATP instead of 36 because they didn't come into glycolysis. Okay, other amino acids can be converted into acetyl-CoA. Others can enter into a part, they can actually be converted into oxaloacetate and enter the Krebs cycle. Okay, so they can enter this process and end up making less than 36 ATP. Glycerol from the fats can be pushed into glycolysis. And then fatty acids can be converted into acetyl-CoA and then go through the process. Okay, so when we teach this ATP production and cellular respiration, we show it using glucose because it goes all the way through and it goes through all the steps and it makes the most ATP. But the other monomers from the foods we eat can be entered into these steps as well. And that's what this is showing here. So let's see. Get my slide to work maybe. Oh no, this happened the other day. Sorry, sometimes my PowerPoint freezes. Good grief, all kinds of crazy things today. Jalen, what am I doing wrong? Fix it.
No clue.
Yeah, sorry, my PowerPoint just starts to go in non-responsive mode. So, and if I stop sharing, then I lose you guys and I can't see you. So anyway, if we go back a slide from this, on mine it's 41. I'm sorry. It says cellular respiration can extract energy from a variety of foods. Okay. That first blank there should be glucose. Okay, glucose often enters our body, like we just talked about, as starch or table sugar, which is sucrose, which is a disaccharide. But energy can also come from fats and proteins, like we saw. And so it talks about intermediate molecules, blah, blah, blah. But basically what happens is those pieces of the amino acids or glycerol or fatty acids can enter into that chain. Okay, that's basically what this slide is saying. Okay, skipping down to 43, it says fatty acids are broken down into 2 carbon fragments, which is the acetyl part of that, and then it can be converted into acetyl-CoA, and enter the blank on slide 43 is the Krebs cycle. Okay, I really wish I could get this to work. Okay, slide 44. Glycolysis is used by virtually every organism on Earth. Okay, we've talked about that. The blank there is under aerobic, A-E-R-O-B-I-C, aerobic conditions. When oxygen is available, cellular respiration follows. We've seen that, okay? If there's no oxygen, we can have anaerobic respiration. And we have talked about this also. You would use, maybe make a note where it says anaerobic, where you would use nitrate or sulfate as a final electron acceptor. Okay, so where it says anaerobic, just make a little note that you would have nitrate or sulfate as a final electron acceptor. Okay, and then on the bottom of that slide, it says, you know, some microbes live where oxygen is limited or not available. And if you think of like, there are some prokaryotes called archaea that are called thermophiles that live at the bottom of the ocean near the thermal heat vents. Okay, there's no oxygen down there, and they would use, I think, sulfate as their final electron acceptor. On the next page, slide 45, it says fermentation takes place in anaerobic conditions. So circle anaerobic indicate that, you know, without oxygen, fermentation can take place. And it says anaerobic organisms rely on fermentation, that's the blank there, for ATP production. And okay, let's take a break there. If you want to continue working on that worksheet, go for it. I'm going to try to get my PowerPoint to restart. Give me a few minutes. Go ahead and take a break, and we'll come back in just a little bit, and hopefully we can get this working. Okay, I think I got it. Let's pull this up. Oops. Let's share with you. Okay. There we go. All right. So fermentation takes place under anaerobic conditions. Okay, so without oxygen, that's what anaerobic means. an in front of a word in science means without. So anaerobic organisms rely on fermentation to help with ATP production. So in fermentation, pyruvate produced by glycolysis enters this metabolic pathway. And there's a few types of fermentation. And it says this pathway, whatever one that pyruvate enters, can be converting pyruvate into either lactate or also called lactic acid, or if it goes into a different type of fermentation pathway, it can make ethanol plus CO2. So there's like this lactic acid pathway of fermentation, and there's others as well. And then there's the other one where you produce ethanol, which is an alcohol, and carbon dioxide. So it's kind of an either or type of a situation. Lactate fermentation, or sometimes called lactic acid fermentation, converts pyruvate to lactic acid. Okay. And so, if you are doing an activity where maybe you're not quite in shape, or maybe you're using muscles that you're not used to using, those muscles may get fatigued and kind of sore. And in those situations, what's happening in your cells is lactic acid fermentation. Okay, so it says during intense muscular activity, blood cannot supply enough oxygen for cellular respiration. Okay, so you're still getting some oxygen, but some of that pyruvate that's being produced needs to go somewhere else because there's just a limited amount of oxygen when you're really working out or working muscles that maybe aren't used to being worked as hard as you're doing at that moment. So muscle cells may briefly, okay, again, for prolonged periods of time, the muscle cells will die without oxygen, but they can briefly use glycolysis and fermentation to generate a few ATP. And what's happening there as far as the metabolic processes, and this is pretty generalized, but you have glycolysis occurring. You get the glucose broken into 2 pyruvate, you get your 2 ATP. But what is really happening here, this 2 ATP we know isn't giving us very much energy. I mean, it is, but compared to 32 that are coming from the electron transport chain, it's not a whole lot. But one of the benefits of lactic acid fermentation, when you're really working out and getting the muscle burn and all of that kind of stuff, is this regeneration of NAD+. It's basically helping you empty out the shuttles, the Ubers, to get them back to pick up more electrons. Okay, because remember with the electron transport chain, that keeps the oxygen, keeps the process funneling electrons through. Okay, so you not only get ATP produced, which is just a little bit, but you also regenerate those NADH so you can keep the pathway moving. Okay, keeps the metabolism going, so you don't have that backup of electrons. Okay, because when you're completely without oxygen, what kills you is that backup of the electrons. These keep part of that pathway going. It kind of supplements in that way. Okay, so says this pathway regenerates or if you like the word recycles, NAD plus by using the electrons and the hydrogens to convert pyruvate to lactate. So I guess I skipped that part. So what it does is in glycolysis, it produces the NADH. It plucks those high energy electrons out and puts them on NADH. And normally that NADH would go to the electron transport chain, but here it can't because there's insufficient oxygen. So the NADH then drops off its electrons here to help produce lactate or lactic acid. Okay, and lactic acid, well, I'll write that on the next slide. The lactic acid is what's going to cause the muscle fatigue and soreness. Okay, and so like if you've ever, worked a muscle hard and you've gotten that fatigue and they feel like jelly and, it's maybe sore for a few days. The reason that those muscles are sore for a few days is it takes the liver that long to basically detoxify the lactic acid and pull that out of the system. But lactic acid fermentation can really help in short-term periods of time and for rapid activities. Okay, so it's kind of like a helpful backup to really, allow your body to exert more effort than it normally could. The other type of fermentation is alcoholic fermentation. And again, this would be under anaerobic conditions. Oh, BIC. Oh my gosh. Okay, and let me just another geek out moment here. In about 15 seconds with insufficient oxygen, the muscle will switch to fermentation, or it will activate that fermentation. So after about 15 seconds, it's like, okay, yep, let's start fermentation. Okay, so that's the one type. That's what our cells do. Okay, when we're doing high demanding activities or activities that maybe our muscles aren't kind of conditioned to do. And the more conditioning you do, if a person was start out to say, okay, I'm going to go, I'm going to condition myself to run a marathon, and they've never run before, they're going to start out slow, right? And they may only run a mile or a half a mile and they're super sore. But your muscles get conditioned. And as you get in better shape, your oxygen transport gets more efficient. And several processes happen more efficiently. So that way, in a year from now, you might be able to run 10 miles and you don't get sore at all, kind of thing. Okay, so switching over to anaerobic alcoholic fermentation. Our cells do not do this, right? So when we're working out or we get a little anaerobic, we don't produce our own alcohol. These would be things like yeast and other microbes that do this. Alcoholic fermentation involves pyruvate. Instead of being converted to lactic acid, it's going to be converted to, like we saw in the other slide, ethanol and carbon dioxide. Okay, and again, it's going to take that NADH from glycolysis that has those high energy electrons and convert it. It's going to pluck them out and produce NAD plus, which the same kind of thing regenerates that so it can go back to glycolysis and pick up more high energy electrons and continue the process. Okay, so same kind of thing. Those high energy electrons are taken to pieces of pyruvate to produce ethanol, which is a 2-carbon molecule, and it plucks one off, and those are lost as carbon dioxide. So if, let's see, was it your guys' class where we had the yeast and you had to smell it in the lab? I think it was. Let's see, did my video quit? Oh, there you are. You guys were all paused, like staring down and not moving. It's like, what happened to them? Okay, there you are. So did you guys?
That connection is unstable.
Okay. Were you guys the ones that had to smell the yeast when we do in the microscope labs? Do you remember that? Okay. That yeast, so that was the yeast that you buy in the little container that you add to your dough to make bread or cinnamon rolls or whatever. Okay. And I think you guys said it smelled like vanilla. But when you are raising some bread and things like that, you have that typical bread dough odor. Okay. That kind of smell, that smells like ethanol. What you're smelling is ethanol. Okay. And the reason that you put yeast in your bread dough is to not necessarily produce the ethanol, but to produce the carbon dioxide. Okay. The carbon dioxide that the yeast produce during the fermentation process is what raises the bread. Okay. Those gases raise the bread during the baking process, and that's what gives you that light, airy texture. Okay. The alcohol, the ethanol in there, though, cooks out, and that's why you don't get drunk by eating bread, is because the ethanol kind of burns off. But it's the carbon dioxide that raises the bread and gives the bread that texture. Fermentation has played an important role in our diet. So obviously, fermentation is used to make wine and beer, thanks to yeasts. And then also baking bread. Like I said, the CO2 gas rising gives the bread or whatever you put the yeast in that airy texture. Okay. We also use fermentation to make soy sauce. It's fermented soybeans. And then there's cheeses that are used, or fermentation is used to make cheeses as well, and several other different types of products that people added yeast and things like that to their food long before they even knew what fermentation was. Okay, so that is the end of our notes for chapter 8. And I wanted to just really quick, because I don't think we'll do this lab on Monday next week, but I wanted to show you this little demonstration of fermentation. So we'll just watch the video here instead of actually doing the lab next week.
As a Navy SEAL, I struggle with PTSD and suicidal thoughts. They threw every pill at me, and nothing worked. Until I tried psychedelic therapy. It helped me to heal, to get my life back. Veterans deserve access to this breakthrough treatment.
Today on Sci Guys, fermenting yeast and sugar. Welcome to Sci Guys. I'm Ryan.
And I'm Teresa, and today we're looking at fermentation with sugar and yeast.
We'll be producing gas, and it won't be me this time. This is a viewer-requested episode. Thank you to this viewer for recommending this episode.
Fermentation is a chemical breakdown of substances by bacteria, yeast, or other microorganisms
usually involving the production of gases and heat.
The equipment and ingredients you're going to need for this episode includes multiple bottles, they can be glass or plastic,
Sugar,
yeast,
balloons,
a funnel,
measuring utensils.
and warm water.
We're not working with anything hazardous in this episode, but we still recommend gloves, goggles, and an apron or lab coat to protect from spills and splashes.
The first step in our experiment is to add all your ingredients into the bottles. Using a funnel, add 1 package or 2 1/4 teaspoons of yeast. Now we're going to add the sugar. In the first bottle, we're not going to add any sugar. In the second bottle, we're going to add 1 teaspoon. In the third bottle, 2 teaspoons. And in the fourth bottle, 3 teaspoons. With all your dry ingredients in the bottles, add 1 cup of warm water into each bottle.
Okay, so this bottle, of course, is going to be the control because there's no sugar. And then each one of these is your experimental treatments, 1x, 2x, and 3x the amounts of sugar.
With all the ingredients in your bottle, cover your bottle with your thumb or put the lid back on and give it a gentle mix.
With all the ingredients in the bottle, put 1 balloon on each bottle spout and leave them somewhere warm to sit for an hour.
Over the next hour or two, the balloons will gradually grow in size. After a few hours, you'll notice the balloon that was covering the bottle that had the most sugar in it grew to a larger size. Let's look at this experiment a little closer. Yeast is a fungus, and there are about 160 known species of yeast. Yeast is very small. 1 gram holds around 25 million cells and has been used for thousands of years to help bread rise. But how does a fungus help bread rise? It has to do with what yeast eats, how they metabolize that food, and what waste products are released. Yeast feeds on sugars and starches. When the yeast consumes the sugar in our bottle, it breaks it down to be used as energy. But like all living beings, waste products are produced when metabolizing food. When the yeast metabolizes the sugar, the waste produced is carbon dioxide gas and ethanol. The carbon dioxide gas floats out of the liquid into the bottle, producing bubbles, and eventually floating up into the balloon. As the yeast consumes more sugar and produces more carbon dioxide, the pressure inside the bottle and balloon increases. This pressure presses on the walls of the balloon, causing it to expand and grow. The equation for this reaction looks like this. The reactant sugar, sucrose, is on the left, and the products, ethanol and carbon dioxide, are on the right. The bottles with greater amounts of sugar have more food for the yeast to consume, which results in larger amounts of carbon dioxide produced and a larger balloon on the top of the bottle.
That's it for fermentation. Thanks for watching.
If you enjoyed this episode, consider supporting our next video by becoming a patron on Patreon. The link is down below. And subscribe for future episodes.
And if you have any suggestions for future experiments,
Okay, so that's one of the labs that we had normally done, but I think the other ones that we're going to do instead are going to be more fun. But we would actually use a piece of string around the balloons and measure the expansion of the balloons and things like that. Okay, so that's fermentation. It occurs when there's no oxygen present, or like with those microbes that we just saw, the yeast, there may be oxygen present, but that's just their preferred way of metabolizing. Okay, so as far as assignments, there'll be chapter 8 in mastering. Okay, that's due Sunday. And the worksheet from the electron transport chain that you guys were just working on, and I saw some of you guys have uploaded that already. And then the third thing is this case study, and it will be due on Friday. And this is a case study of an actual event that happened in 1982. And I remember it. Yes, I'm that old. I remember when this happened. It was in Chicago, and I grew up 100 miles west of Chicago, and people were kind of freaking out when this happened, and until they figured out what was causing it. So this is an actual event, and it ties into cellular respiration. Okay, cellular respiration was altered, and that's why these people died. Okay, so as you're going through the case study, I would suggest writing the answers on a separate piece of paper or typing them out in a separate document. Excuse me, but part one talks about some of the symptoms and basically gives some background on what's going on. Oh, excuse me, what's going on. And then there's a couple questions here. And then down here, just to kind of, so we're all on the same page here, it talks about the autopsy report of some of the people that passed away. Their immediate cause of death was hypoxia. And so that's what they call it when a person is not getting enough oxygen. They looked at heart, lung, kidney, liver, and saw all kinds of cell death, massive cell death. They stained some of the cells and they saw there was a bunch of damage to the mitochondria. And then they said oxygen levels were 110 millimeters of mercury, where normally it should be 75 to 100. So something interesting going on there. And then it also gives so the word metabolite here is basically like pieces of the metabolic pathways. Okay, so don't get tripped up on that word. It just means pieces of the metabolic pathway. So if you were to take out glucose, they looked at patient levels, pyruvate, which is a product of glycolysis, NAD plus, and NADH. Okay, so this is like a critical thinking exercise where I want you to think about and apply the information that you've learned from cellular respiration and like what it means to have low numbers of NAD plus, what it means to have high numbers of NADH. Think about things that we've talked about over the last couple of days, especially today, about what could be causing these things to happen. Okay, and it'll ask you some questions here about that, and then kind of finish up some of the information here. Okay, so this is due on Friday. If you're working through this and you're like, I don't get this part, please send me an email. I can definitely try to help point you in the right direction. So chapter 8 in mastering, the case study there, and then the electron transport chain worksheet that we worked on in class, get that uploaded. And then don't forget, we're meeting in person to do labs on Monday. And if there's someone, I can't remember if someone was printing the labs. Was that you, Alexis? Did you print labs? I'm sorry, I can't hear you.
Wait, printing the labs, what do you mean?
Do you have the lab manual or do you print them off?
No, I still am online.
Okay, I will post the lab pages that we need for Monday in case somebody needs to print them ahead of time if they don't have the lab manual. Okay. All right, we will see you guys on Monday.